Beneath the quiet surface of our everyday interactions, love and feelings operate as the most sophisticated biological signaling systems ever evolved. This complex web of neurochemistry ensures our survival and binds us together in ways that are often invisible to the naked eye. Oxytocin, a small peptide produced in the hypothalamus, acts as a primary chemical messenger that fosters social bonding and parental care across the mammalian kingdom. If you find value in these quiet insights, subscribing helps us continue exploring the mechanics of life together. While we often describe affection as a purely poetic experience, it functions primarily as a survival mechanism designed by evolution to encourage cooperation and protection of the vulnerable. In the brain, the ventral tegmental area and the caudate nucleus light up during intense feelings of attraction, flooding the system with dopamine. This reward-based architecture mirrors the same pathways activated by basic needs like hunger or thirst, suggesting that emotional attachment is a biological imperative rather than a luxury. The architecture of affection is visible even at a microscopic level, where specific receptors in our cells are tuned to receive these emotional signals. Research published in the journal Nature indicates that these chemical bonds can influence everything from our immune system's efficiency to the speed at which physical wounds heal. We are beginning to understand how these internal states transition from tiny neurotransmitters into the macroscopic reality of human history and culture. By examining the physiological reality of our hearts and minds, we find that what we feel is a meticulously calibrated response to our environment. This exploration will trace the journey of a feeling from its origins in the primitive brain to its expression in our modern lives, providing a steady stream of relaxing facts for your bedtime listening. Biological anthropologist Helen Fisher suggests that romantic love is not a single emotion but a suite of three distinct brain systems: lust, attraction, and attachment. Each stage utilizes a unique chemical profile to drive specific evolutionary behaviors essential for species survival. Lust is primarily governed by the hypothalamus, which stimulates the production of testosterone and estrogen to initiate the search for a partner. As interest deepens, the brain transitions into the attraction phase, often referred to as the romantic or infatuation stage. Here, the reward system is flooded with dopamine and norepinephrine, creating a state of focused attention and high energy. This surge can lead to a decreased need for sleep and a suppressed appetite, as the brain prioritizes the pursuit of the beloved above basic maintenance. Long-term stability eventually gives way to attachment, a stage dominated by the neuropeptides oxytocin and vasopressin. These chemicals are instrumental in fostering feelings of security and social comfort, allowing a couple to remain together long enough to cooperate in raising offspring. Before any of these complex stages can take root, however, the brain performs an incredibly rapid assessment of every person we encounter. Research published in the journal Psychological Science indicates that it takes only 1/10 of a second for a person to form a judgment about another's attractiveness and trustworthiness. This millisecond-scale processing occurs within the amygdala and the posterior cingulate cortex, areas of the brain involved in emotional evaluation and social decision-making. During this initial encounter, the visual system prioritizes certain facial features that provide cues about health and genetic fitness. Subconscious attention is directed toward bilateral symmetry, which is often interpreted by the brain as a sign of developmental stability. Can you recall a time when you felt an immediate, unexplainable sense of comfort upon meeting someone for the first time? This gut feeling is often the result of the ventral striatum activating in response to perceived facial beauty and social compatibility. Even the dilation of a stranger's pupils is processed subconsciously. Wider pupils are often perceived as more attractive because they signal physiological interest and arousal. The brain also evaluates major histocompatibility complex genes through subtle olfactory signals, ensuring we are compatible on a molecular level before we even begin to learn a person's name. Every microexpression and facial contour is mapped against internal templates of safety and desirability in less time than it takes to blink. These invisible mechanisms illustrate how deeply our interpersonal connections are rooted in involuntary biological processes. Once these initial templates are established, the brain pivots toward an intense, highly focused state governed by the ventral tegmental area, or VT. Located deep within the midbrain, the VTA functions as one of the primary engines of the dopaminergic reward system. It is here that neurons synthesize dopamine and distribute it toward various cortical regions, signaling that a specific stimulus is of the utmost importance. When a person is in the early, obsessive stages of romantic love, the VT fires with a frequency and intensity that mirrors the fundamental survival drives of thirst or hunger. This activity is not merely a reaction to a pleasant experience. It is a primal signaling system that prioritizes the beloved above almost all other environmental stimuli. Adjacent to this hub of activity sits the caudate nucleus, a large, C-shaped structure that plays a critical role in the integration of sensory data and the coordination of movement toward a goal. Research utilizing functional MRI scans has consistently shown that when an individual views a photograph of their partner, the caudate nucleus illuminates with significant metabolic activity. This part of the brain is intrinsically linked to the reward anticipation system, helping to map out the physical and mental actions required to secure a desired outcome. The caudate acts as a bridge between the raw chemical impulse of the VTA and the sophisticated planning required to pursue a relationship. While many associate dopamine exclusively with the sensation of pleasure, its primary function in the context of human attachment is actually the regulation of motivation and wanting. In neuroscience, this is often distinguished from liking. While liking is a hedonic response to a stimulus already present, wanting is the intense, focused drive to obtain a reward that is not yet fully secured. This is why the early stages of love can feel so incredibly energetic and, at times, exhausting. The brain is effectively locked into a state of high-alert craving, channeling all available cognitive resources toward the pursuit of the other person. This elevated dopamine level sharpens our focus, often leading to what psychologists call intrusive thinking, where the image of the beloved occupies the mind for the vast majority of waking hours. The biological profile of this state bears a striking resemblance to the brain chemistry observed in individuals under the influence of powerful stimulants. When scientists compare brain scans of those in the throes of new love with those who have used substances like cocaine, the similarities in the activation patterns of the reward circuitry are undeniable. Both states involve a massive flooding of the nucleus accumbens with dopamine, which creates a loop of reinforcement that can be difficult to break. It is a strictly chemical reality that the brain in love is a brain in a state of healthy addiction, seeking its fix through social proximity and emotional validation. This comparison provides a scientific basis for why the loss of a relationship can trigger physical withdrawal symptoms similar to those found in clinical dependency. This system is inherently goal-directed, designed by evolution to ensure that humans remain focused on a specific partner long enough to establish a pair bond. The intense physiological arousal and the narrowing of interest serve the purpose of narrowing one's social field to a single, vital point of contact. This drive is so powerful that it often bypasses the executive functions of the prefrontal cortex, leading to the blindness often associated with new romance. Can you recall a time when your own focus became so narrow that the rest of the world seemed to fade into the background? This state of heightened attention is the direct result of the caudate nucleus and the VTA working in tandem to ensure that the pursuit of connection remains the brain's highest priority. This chemical surge is largely driven by norepinephrine, a neurotransmitter that doubles as a hormone to mobilize the brain and body for action. It is synthesized from dopamine within the brain and the adrenal glands, and its release marks the transition from simple interest to physiological obsession. When you are near someone you are falling for, your sympathetic nervous system enters a state of hyperarousal, causing your pupils to dilate to let in more visual information about the other person. This dilation is an involuntary response that makes the eyes appear darker and more inviting, often subconsciously signaling interest back to the partner through subtle nonverbal cues. As the heart rate climbs, the body's metabolic rate increases, and the liver releases glucose into the bloodstream to provide an immediate energy boost. This is why many people in the early stages of love report feeling a diminished need for food or sleep. The body is essentially running on a high-octane chemical fuel. This heightened state directly influences the amygdala, the brain's emotional processing center, which becomes more sensitive to every nuance of the partner's behavior. The intense focus provided by norepinephrine is what allows the rest of the world to slip away during a conversation, creating a sense of intimacy that feels nearly impenetrable to outside interference. This attentional narrowing ensures that the brain filters out distractions to focus entirely on the source of the reward. At the same time, this arousal acts as a powerful catalyst for memory. The brain uses these spikes in norepinephrine to mark specific events as significant, leading to the formation of exceptionally clear and lasting memories. Do you find that certain songs or smells immediately transport you back to a specific moment from your past? This deep encoding is a result of long-term potentiation in the hippocampus, which is why you might remember the exact temperature of the air or the specific pattern on a tablecloth from a first encounter years later. This physiological state is so potent that it can be easily misidentified through the process of misattribution of arousal. When the body experiences a high arousal state from an external source, like the adrenaline from a roller coaster or the fear from a suspenseful movie, the brain often looks for the most logical explanation in the immediate environment. In a landmark 1974 study by Dutton and Aaron, researchers found that men crossing a high, swaying suspension bridge were more likely to find an interviewer attractive than those on a low, stable bridge. The physiological stress of the height was subconsciously relabeled by the brain as romantic interest. This suggests that the context in which we meet someone can significantly influence our initial feelings of chemistry. Even a shared frightening experience or a high-intensity workout can artificially boost the perceived level of attraction between two people. This crossover between fear and attraction highlights how closely our primitive survival instincts are interwoven with our most sophisticated emotional experiences. While the brain is bathed in the rewarding glow of dopamine, a simultaneous and dramatic decline occurs in the levels of serotonin circulating through the neural pathways. This specific chemical shift is responsible for the intense, often intrusive preoccupation that defines the early months of a romantic bond. Research conducted at the University of Pisa has shown that the serotonin levels of people in the initial stages of attraction are significantly lower than those in a neutral control group. These depleted levels are often indistinguishable from the neurochemical profiles found in individuals diagnosed with clinical obsessive-compulsive disorder. This shared chemical state explains why a new lover can become the singular focus of an internal monologue, appearing in thoughts hundreds of times a day without conscious consent. The evolutionary purpose behind this temporary state of obsession is deeply rooted in the preservation of the species. By lowering serotonin, the brain essentially creates a state of psychological tunnel vision that ensures all available cognitive and physical energy is directed toward a single partner. In a primitive environment, the process of selecting a mate and establishing a bond required an immense amount of focus and resource allocation. This biological fixation acted as a safeguard, preventing the individual from being easily distracted by other potential mates or environmental interests until a stable pair bond was formed. Have you ever looked back at an old relationship and wondered why you were so completely consumed by every small detail in those first few months? This is the result of your brain prioritizing the survival of the bond over the balance of your own moods. During this period, the brain's ability to maintain emotional equilibrium is often compromised, leading to the characteristic highs and lows of early love. The lack of serotonin makes the mind more sensitive to the presence or absence of the partner, mirroring the way a person with OCD might feel a sense of unease if a specific routine is interrupted. This neurochemical imbalance is often accompanied by elevated levels of cortisol, the body's primary stress hormone, which further heightens the sense of urgency and emotional intensity. It is a demanding state for the body to maintain, requiring a high metabolic cost to keep the nervous system in such a heightened state of alert. Fortunately, the human brain is designed to return to a state of homeostasis once the initial goal of bonding has been achieved. Science indicates that these serotonin levels typically begin to normalize within 12 to 18 months of the relationship's onset. As the chemical baseline returns, the intrusive, obsessive thoughts begin to lose their grip, allowing the individual to regain their sense of self and broader perspective. This transition does not signal the end of the affection, but rather the maturation of the emotion into a more stable and sustainable form. The frantic energy of limerence fades into the steady, quiet security of companionate love, which is far less taxing on the body's internal chemistry. This stabilization is vital for the long-term health of both the individual and the partnership, as it allows for a return to rational judgment and the restoration of a balanced life. The transition from intense passion to steady companionship is governed by the quiet work of the hypothalamus. Within this small but mighty region of the brain, a peptide hormone called oxytocin is synthesized, waiting for the precise signal to enter the bloodstream. It is often referred to as the molecule of connection, yet its biological function is far more complex than simple affection. Once produced in the paraventricular and supraoptic nuclei, It travels to the posterior pituitary gland for storage and eventual release. When you experience a gentle touch or a warm embrace, the brain triggers a surge that floods the system with a sense of calm and trust. This hormonal response serves as the biological foundation for human bonding, particularly during the most vulnerable moments of life. During childbirth, a massive surge of oxytocin facilitates uterine contractions and later drives the milk ejection reflex during nursing. It is a biological imperative designed to ensure the survival of the offspring through an unbreakable emotional link. However, the influence of this molecule extends well beyond the maternal bond. It acts as a neuromodulator, fine-tuning how we perceive and respond to the social world around us in every interaction. Recent research conducted throughout 2024 and 2025 has deepened our understanding of the dual nature of oxytocin. While often celebrated as a cuddle chemical, contemporary studies have revealed its role in sharpening the boundaries between us and them. This molecule does not merely promote universal love, it intensifies in-group bias, making an individual more protective of their immediate circle while potentially increasing suspicion toward outsiders. It is an evolutionary mechanism for protection, ensuring that the resources and safety of the group are maintained against perceived threats. Can you recall a time when your loyalty to a friend felt almost instinctive, even before you had time to think about it? The physiological impact of oxytocin is most notable in the amygdala, the brain's primary center for processing fear and emotional threats. By binding to specific receptors in this almond-shaped structure, oxytocin effectively dampens the neural activity associated with anxiety and social hesitation. This inhibition allows for a significant reduction in the stress response, helping to lower cortisol levels and promote a state of physiological safety. It essentially mutes the alarm bells of the brain, making it possible to form deep connections without the interference of the fight-or-flight response. In the social sphere, oxytocin enhances our ability to read facial expressions and interpret subtle emotional cues from others. This heightened social salience makes interactions feel more rewarding and less taxing on our cognitive resources. It is through this mechanism that the hormone helps bridge the gap between two separate individuals, weaving their experiences into a shared narrative. By reducing the perceived risk of social rejection, the brain allows for a degree of vulnerability that is necessary for a long-term, stable partnership. As we look at the neurobiology of the human heart, we find that oxytocin is the architect of stability. It doesn't just make us feel good, it organizes our social world into a hierarchy of trust and safety. This sophisticated chemical system ensures that once the fire of early infatuation settles, it is replaced by a profound, enduring architecture of mutual support. This transition is not a loss of intensity, but a gain in structural integrity, allowing love to survive the pressures of the external world. While oxytocin is often celebrated for its role in maternal bonding and immediate social warmth, vasopressin operates as its more stoic and protective counterpart. This small peptide hormone, synthesized in the hypothalamus and released by the posterior pituitary, plays a dual role in the human body, managing fluid balance while simultaneously sculpting the neurological landscape of long-term fidelity. In the context of pair bonding, vasopressin is the chemical signal that shifts the focus from the internal glow of attraction to the external reality of partnership maintenance. It is less about the softness of a touch and more about the quiet vigilance required to preserve a shared life over decades. Science has found some of its most compelling evidence for this in the study of voles, specifically the stark contrast between the prairie vole and the montane vole. These two species are genetically similar yet exhibit vastly different social structures that have fascinated researchers for years. Prairie voles are famous for their lifelong monogamy and shared parental duties, a behavior directly linked to a high density of vasopressin receptors in the reward centers of their brains, specifically the ventral pallidum. When a male prairie vole mates, a surge of vasopressin rewards the brain, creating a permanent neurological association between the partner and a sense of well-being. Conversely, the montane vole remains solitary and promiscuous, lacking those same concentrated receptor sites. Researchers discovered that by artificially increasing vasopressin receptor expression in these more solitary rodents using viral vector gene transfer, they could induce pair bonding behaviors that were previously absent from the species' repertoire. This suggests that the capacity for commitment is not just a matter of personality, but a matter of how the brain is physically wired to receive specific chemical signals. Have you ever wondered why some people seem naturally more inclined toward the comfort of long-term stability than others? In addition to bonding, vasopressin is intricately linked to territoriality and what biologists define as mate guarding. In many mammalian species, this hormone triggers a protective instinct that ensures the safety of the partner and the survival of the offspring. It transforms the individual from a solitary actor into a dedicated guardian of the domestic sphere. In humans, this translates to a heightened sense of commitment and a subconscious drive to provide and protect, grounding the relationship in a sense of duty that persists even when the initial excitement of a romance begins to fade. The human application of these findings centers on the AVPR1A gene, which codes for the vasopressin receptor. Geneticists have identified specific variations in this gene, such as the RS3334 allele, which appear to influence how individuals experience and maintain their closest relationships. Scientific observations through 2024 and 2025 have reinforced the idea that people with certain versions of this gene may report lower levels of marital quality or a different perception of bonding. However, biology is rarely destiny. These genetic markers represent tendencies that are filtered through the lens of our experiences and values. Vasopressin provides the evolutionary foundation for the concept of home, acting as the invisible glue that maintains the social fabric and turns a transient connection into a permanent sanctuary. While vasopressin builds the structural architecture of our long-term bonds, the vagus nerve provides the real-time electrical wiring for what researchers call the social engagement system. Often described as the primary highway of the mind-body connection, the vagus nerve is the longest cranial nerve in the human body, stretching from the brainstem through the neck and down into the abdomen. It serves as the physiological foundation for polyvagal theory, a framework established by Dr. Stephen Porges that explains how our nervous system distinguishes between environments of threat and environments of safety. When we are in the presence of someone we love, the ventral vagal pathway is activated, essentially acting as a biological brake on the sympathetic nervous system's fight-or-flight response. This activation allows the body to divert energy away from defensive postures and toward the complex processes of social bonding and emotional communication. Scientists quantify the health and efficiency of this system through a metric known as vagal tone, which is measured by tracking the relationship between your heart rate and your breathing patterns. A high vagal tone is consistently correlated with a greater capacity for emotional regulation and social flexibility, acting as a biological buffer that helps individuals navigate the inevitable frictions of an intimate relationship. Because this nerve influences the muscles of the throat and the middle ear, it even changes how we perceive sound, filtering out low-frequency background noise so we can more easily focus on the melodic frequencies of a partner's voice. The influence of the vagus nerve extends beyond the individual, creating a phenomenon known as physiological co-regulation. When two people are in close physical proximity, their autonomic nervous systems begin to mirror one another, often leading to a synchronization of heart rate variability. This means that a calm partner can quite literally act as an external regulator for a stressed partner, using the silent language of the nervous system to broadcast safety through the vagus nerve. This feedback loop is further reinforced by the intricate connection between the vagus nerve and the muscles of facial expression. When you see a genuine, Duchenne smile from a loved one, your brain processes those micro-expressions and sends an immediate signal down the vagus nerve to slow your heart rate and relax your digestive system. This bidirectional communication ensures that our internal state is constantly being updated by the social cues we receive from those around us. By the start of 2026, clinical applications of vagal nerve stimulation have increasingly highlighted how critical this pathway is for maintaining the pro-social behaviors that allow human societies to flourish. Through this nerve, the abstract feeling of safety is transformed into a tangible, physical reality that sustains our most cherished connections. Shifting focus from the immediate biological mechanics of the nervous system, we look back through deep time to understand why natural selection favored the intense, often overwhelming experience of human love. Evolutionary psychology suggests that love is not merely a social construct or a poetic invention, but a sophisticated survival strategy hardwired into our species. Human infants are remarkably altricial, a term used in biology to describe offspring born in a highly undeveloped and helpless state. Unlike many mammals that can walk within hours of birth, human children require years of constant care and protection. This biological vulnerability created a unique pressure on our ancestors, leading to what researchers call the parental investment theory. Because the cost of raising a human child to maturity is so high, the survival of the offspring was significantly increased when both parents remained together to provide resources and protection. In this context, love evolved as a powerful psychological glue, a commitment device designed to keep pairs bonded long enough to ensure the next generation reached reproductive age. This shift toward pair bonding is actually quite rare in the animal kingdom, occurring in only about 3 to 5% of all mammalian species. The obstetrical dilemma explains why this bond became so critical. As human brain sizes increased and our ancestors began walking upright, infants had to be born earlier in their development to fit through the birth canal. This led to a prolonged period of childhood, necessitating a social structure that could sustain long-term cooperation. Do you remember a moment when the presence of a loved one seemed to wash away your anxieties, making the world feel safer than it did just moments before? This feeling is the result of specific cognitive trade-offs that occur when we fall in love. Neuroimaging studies, such as those conducted by Samir Zeki at University College London, show that when we view a romantic partner, the regions of the brain responsible for social judgment and critical thought, specifically the prefrontal cortex, show a marked decrease in activity. This suspension of judgment is an evolutionary mechanism that allows us to overlook minor flaws and maintain the stability of the bond during the early, critical stages of attachment. Beyond commitment, evolutionary psychology examines how we select partners through the lens of fitness indicators. These are observable traits that signal underlying genetic quality and health. Bilateral symmetry in the face and body is one such indicator, as it suggests a developmental history free from major disease or environmental stress. Our senses also play a silent role in this selection process through the major histocompatibility complex, or MHC. Research has shown that humans are often unconsciously attracted to the scent of individuals whose immune system genes differ from their own. This genetic diversity provides offspring with a more robust immune system, better equipped to handle a wider variety of pathogens. Love also functions as a costly signal, a concept where an individual demonstrates their value through behaviors that are difficult to fake, such as the devotion of time, protection, and shared resources. By prioritizing one individual over all others, the brain signals a high level of reproductive and social investment. Even the grandmother hypothesis fits into this evolutionary framework, suggesting that the long post-reproductive lifespan of human females evolved to provide additional support for the survival of grandchildren, further weaving the fabric of love and kin into the very survival of our lineage. The evolutionary drive for survival extends beyond caregiving into the very chemistry of how we choose our partners, operating through a group of genes known as the major histocompatibility complex, or MHC. These genes are responsible for coding the proteins that help the immune system distinguish between the body's own cells and dangerous invaders like viruses or bacteria. While we might think we are choosing a partner based on their smile or their wit, our noses are busy scanning for a specific genetic signature that complements our own. This molecular identification system is so precise that it can influence our social preferences and the intensity of our romantic attractions. In the mid-90s, a landmark study often referred to as the sweaty t-shirt experiment provided some of the first empirical evidence for this olfactory attraction. Researcher Klaus Wedekind asked a group of women to sniff cotton t-shirts that had been worn for 2 consecutive nights by men they had never met. The results showed that the women consistently ranked the scents as more pleasant when the man's MHC genes were significantly different from their own. This suggests that humans possess a sophisticated biological radar for immune system diversity, even if we are not consciously aware of the data we are processing. Have you ever noticed how the natural scent of someone you loved felt inexplicably comforting, even if it was not a traditional perfume or cologne? This preference for dissimilarity is a calculated biological strategy to ensure the health of future generations. When 2 parents with diverse MHC profiles reproduce, their offspring inherit a broader, more robust immune system capable of recognizing a wider array of pathogens. This genetic variety, often called heterozygosity, provides a distinct survival advantage in an environment filled with evolving diseases. By selecting a mate whose immune strengths cover our own weaknesses, we are essentially building a protective shield for our children before they are even conceived. The nuances of this system become even more fascinating when we consider how modern medicine can inadvertently interfere with these ancient signals. Research has shown that hormonal contraceptives, which alter a woman's hormonal state to mimic pregnancy, can actually flip these scent preferences. When the body believes it is already pregnant, the biological drive shifts away from finding a genetically compatible mate and towards seeking the protection of close kin. Consequently, some women on the pill may find themselves more attracted to men with MHC profiles similar to their own, potentially altering the chemical chemistry of a relationship if they later stop taking the medication. The vomeronasal organ and the olfactory epithelium work in tandem to process these complex chemical messages, sending signals directly to the limbic system, the brain's emotional center. This bypasses the rational cortex entirely, explaining why attraction often feels like an undeniable, visceral force that we cannot quite put into words. It is a silent dialogue between two immune systems, a microscopic vetting process that has been refined over millions of years of mammalian evolution. Even in our modern world of digital dating and curated profiles, the primal necessity of genetic compatibility remains a quiet, powerful architect of human intimacy. This physical geometry often finds its most precise expression in the concept of bilateral symmetry, a mathematical balance between the left and right sides of the face. When we look at a person, our visual cortex performs a lightning-fast calculation of these proportions, seeking out the elusive golden ratio, or phi, which sits at approximately 1.618. This ratio governs the spacing between the eyes, the length of the nose, and the width of the smile, serving as a silent blueprint for what we perceive as aesthetic perfection. Biologically, this preference is far more than a superficial pursuit of vanity. It is an assessment of developmental stability. During the intricate process of gestation and childhood growth, the human body must navigate a gauntlet of environmental stressors, including pathogens, toxins, and nutritional deficiencies. High degrees of symmetry suggest that an individual possessed the genetic robustness to withstand these pressures, maintaining a steady course of development despite external disruptions. Conversely, fluctuating asymmetry serves as a subtle biological warning sign, signaling that the developmental process was perhaps compromised by illness or genetic mutations. While we rarely consciously notice a slightly lower ear or a minor tilt in a jawline, the subconscious mind is incredibly sensitive to these deviations. This aversion to asymmetry evolved as a protective mechanism, helping our ancestors identify partners who were likely to possess the healthiest immune systems. Have you ever noticed how your own perception of someone's face changes the more time you spend looking into their eyes? This phenomenon is closely tied to the concept of facial averageness, a counterintuitive finding in evolutionary psychology that suggests the most attractive faces are those that represent the mathematical mean of a population. Research using composite imaging has consistently shown that when dozens of individual faces are digitally blended together, the resulting image is almost always rated as more attractive than any of the single faces that comprise it. This happens because the averaging process smooths out idiosyncratic irregularities, creating a face that feels familiar, balanced, and free of extreme mutations. It suggests a diverse genetic heritage, which often correlates with a more versatile and resilient immune response. Beyond the structure of the bone and skin, the eyes offer their own set of high-contrast signals that bypass the rational mind entirely. The limbal ring, that dark circular border where the iris meets the white sclera, acts as an honest indicator of youth and cardiovascular health. Because these rings are darkest and most defined in early adulthood and naturally fade with age or certain medical conditions, a thick, prominent limbal ring is a powerful, if quiet, attractant. This visual dialogue is further intensified by the behavior of the pupils, which dilate involuntarily during moments of genuine interest or arousal. This response is controlled by the autonomic nervous system, making it impossible to fake and providing a raw, honest window into a person's internal state. In the 16th century, women even used extracts from the belladonna plant to artificially dilate their pupils, intuitively understanding that large pupils signaled a profound, though chemically induced, attraction. These intricate physical markers, from the precision of a facial ratio to the involuntary expansion of a pupil, weave together to form the silent language of beauty, ensuring that our deepest feelings are always rooted in the elegant, unyielding laws of biological physics. The physical culmination of this attraction often finds its most intense expression in the press of the lips, an act scrutinized by a specialized field of study known as philematology. While we often think of our hands as our primary tools for tactile exploration, the lips possess a significantly higher density of nerve endings than even the most sensitive fingertips. This biological sensitivity transforms a simple touch into a high-bandwidth data stream for the brain, processing heat, pressure, and texture with staggering precision through a high concentration of Meissner's corpuscles. Within the somatosensory cortex, the region of the brain that maps our sense of touch, the space dedicated to the lips is disproportionately large, reflecting their status as one of our most acute sensory interfaces. This proximity allows for a deep, subconscious assessment of a partner through the exchange of chemical information. During a passionate kiss lasting roughly 10 seconds, research published in the journal Microbiome has shown that partners can exchange as many as 80 million bacteria. While this might sound unappealing, this shared microbiota actually serves as a biological handshake, potentially priming the immune system and assessing genetic compatibility through the major histocompatibility complex. Furthermore, male saliva contains trace amounts of testosterone, which can be absorbed through the mucosal membranes of the mouth. Over time, this transfer can increase the libido of a partner, subtly influencing the hormonal balance of the interaction. It functions as a chemical taste test, an ancient mechanism designed to determine if a pairing is viable at a microscopic level. Have you ever wondered why some memories of a first kiss remain so much more vivid than other significant life events? This clarity is likely due to the surge of dopamine and oxytocin that floods the system, creating a neurological imprint that signifies importance. Yet, despite its biological utility, romantic kissing is not a universal human behavior. A landmark study from 2015, which remains a primary reference for anthropologists in 2026, found that only about 46% of the 168 cultures surveyed engaged in romantic sexual kissing. Many societies find the practice uncomfortable or even unhygienic, yet they almost always maintain close contact equivalents. In cultures where lip-to-lip contact is absent, behaviors like the Malay kiss or the Inuit kiss involve pressing the nose against a partner's cheek and inhaling deeply. This sniff kiss serves the same evolutionary purpose, allowing individuals to process the pheromones and natural scents that signal health and genetic diversity. Whether through the press of lips or the sharing of breath, these rituals ensure that our bodies remain in constant, quiet communication with those we hold close, turning the simple act of proximity into a complex, life-sustaining dialogue. Deep within the architecture of our skin lies a specialized system of neurons designed specifically to register the emotional quality of a caress. These are the C-tactile afferents, or CT fibers, a class of unmyelinated nerve endings found exclusively in the hairy skin of the body, distinct from the glabrous skin of the palms or soles. Unlike the fast-conducting A-beta fibers that provide immediate feedback on pressure, vibration, or texture, CT fibers are slow and methodical, transmitting signals at a rate of approximately 1 meter per second. This deliberate pace reflects their unique purpose. They are tuned to the specific velocity and temperature of a human hand. Can you recall a time when a simple hand on your shoulder instantly lowered your heart rate during a moment of stress? This physiological shift occurs because the activation of CT fibers triggers the release of oxytocin while simultaneously inhibiting the hypothalamic-pituitary-adrenal axis, reducing the circulation of cortisol. When this system is understimulated, humans experience a phenomenon known as skin hunger or touch starvation. Long-term touch deprivation is linked to increased levels of anxiety and a measurable weakening of the immune system as the body loses a primary external regulator of its internal homeostasis. In clinical settings, the lack of tactile stimulation has been shown to alter the development of the brain's social circuits, highlighting that touch is a biological necessity rather than a secondary comfort. The evolutionary roots of this system are visible in the allogrooming behaviors of non-human primates, where picking through fur serves as a vital social currency that maintains group cohesion and reduces conflict. In humans, while we have lost the dense pelage of our ancestors, we have retained the neural infrastructure that makes grooming-like behaviors, such as hair brushing or gentle stroking, deeply rewarding. This system ensures that we remain tethered to our social groups through a reinforcing loop of neurochemical rewards. By prioritizing these slow-acting signals, the brain differentiates between the functional touch of a tool and the meaningful touch of a companion, effectively turning the skin into a social organ. This constant, quiet monitoring of our physical boundaries allows the body to maintain a sense of safety and belonging within the complex social landscapes we navigate every day. If you find this exploration of our hidden biological networks fascinating, consider following for more deep dives into the science of human connection. The way our nervous system translates a physical stroke into an emotional anchor remains one of the most elegant proofs of our inherently social nature. The presence of a loved one does more than just soothe the mind. It actively recalibrates the rhythmic machinery of the body. When long-term partners sit in close proximity without even speaking, their autonomic nervous systems begin to mirror one another in a silent, invisible dialogue. This physiological entrainment means that heart rates fluctuate in tandem and respiratory patterns drift into a shared cadence. Research indicates this is not merely a byproduct of being in the same room, but a sophisticated subconscious adjustment to the presence of a trusted individual. Beyond the mechanical rhythm of the heart, our brains engage in an intricate dance of electromagnetic alignment. Throughout 2025, advanced neuroimaging studies have expanded our understanding of neural coupling, a phenomenon that occurs during moments of deep narrative sharing or conversation. When one partner recounts a vivid experience, the listener's brain activity often mirrors the speaker's with a slight temporal lag. In the most profound instances of connection, this lag disappears, and the listener's neural responses begin to anticipate the speaker's upcoming words. This suggests that the two brains are no longer processing information in isolation but are functioning as a single, predictive circuit. The power of this biological bond manifests most clearly in the modulation of physical pain and stress. When an individual faces a painful stimulus, the brain's threat response centers, such as the anterior cingulate cortex and the hypothalamus, typically flare with intense activity. However, if they simply hold the hand of a romantic partner, this neural activation is significantly dampened. This phenomenon, central to social baseline theory, implies that the brain perceives a partner as a literal extension of its own survival resources. By offloading the metabolic cost of threat management to the relationship, the individual experiences less distress and a more efficient recovery. Do you remember a time when the mere presence of someone special made a difficult moment feel suddenly manageable? This is the tangible result of a dyadic biological system. Scientists now view such pairs not as two independent organisms, but as a single regulatory unit where one person's physiology serves as a stabilizing force for the other. This interconnectedness allows for a shared emotional load, where the fluctuations of stress are literally distributed across two nervous systems. This mutual regulation is the biological foundation of intimacy, where the boundaries of the self expand to include the physiology of the partner. In this state, the health of one person becomes intrinsically linked to the regulatory capacity of the other. It is a resilient feedback loop that promotes cardiovascular stability and immune function over a lifetime. This silent communication between bodies proves that we are never truly solitary in our experiences. We are designed to be part of a larger, synchronized whole. The architecture of our hearts and minds is built for this level of integration. While we often think of love as a psychological state, it is fundamentally an organizational principle for our biological existence. Our bodies are constantly scanning for the signals of safety and resonance provided by another, using that input to maintain internal balance and long-term health. The intense, high-voltage electricity of new love is essentially a biological loan that must eventually be repaid to maintain the equilibrium of the organism. Within the first year to 18 months of a relationship, the brain's ventral tegmental area begins to dampen its frantic output of dopamine. This specific neurotransmitter is responsible for the obsessive focus and the sleepless nights often seen in the early stages of romance. While it feels like a loss of intensity, this physiological cooling is a vital protective mechanism for the human nervous system. High levels of dopamine and norepinephrine act similarly to a chronic stress state, keeping the body in a prolonged state of arousal that is metabolically expensive to maintain over long durations. As the frantic activity of the reward system recedes, a quieter and far more durable network begins to take over the primary functions of the bond. Neuroimaging studies, including those documented in the journal Nature, show that in couples who have transitioned into long-term stability, the focus of neural activity moves toward the globus pallidus and the ventral pallidum. These regions are densely packed with receptors for oxytocin and vasopressin, the chemicals primarily responsible for the deep sense of security and pair bonding. This shift represents a fundamental change in the brain's objective, moving from the pursuit of a reward to the preservation of an established connection. It is a biological pivot from the thrill of the chase to the quietude of the sanctuary. This second stage, frequently categorized as companionate or compassionate love, offers profound energy-saving benefits for the human body. When we exist in a state of secure attachment, our baseline physiological arousal is significantly lower, allowing our heart rate to remain steady and our respiratory system to function with greater ease. Can you recall a specific moment when the simple presence of a partner seemed to physically lower your internal tension after an exhausting day? This phenomenon is the tangible result of the brain offloading its regulatory duties onto the relationship itself. Because the partner is perceived as a reliable source of safety, the body no longer needs to expend metabolic energy on hypervigilance or social anxiety. While popular culture often views the fading of the initial honeymoon phase as a tragedy, the science of psychology suggests it is actually a sophisticated maturation of the bond. The transition signifies that the relationship has moved from a state of neurochemical instability to a state of structural permanence. In this phase, the prefrontal cortex, the region responsible for complex decision-making and long-term planning, becomes more integrated with the emotional centers of the brain. This allows for a reduction in amygdala activity, meaning the brain effectively switches off its threat detection when interacting with a long-term partner. Instead of two individuals operating in a state of high alert, the pair bond begins to function as a single, more efficient biological unit. This maturation allows the couple to navigate external stressors with a shared cognitive load, essentially sharing the burden of existence. The transition to compassionate love is not the end of the flame, but rather the moment the fire moves from the kindling to the hearth, providing a steady warmth that can sustain a lifetime. The architecture of the human brain is fundamentally social, built through the intricate biological feedback loops defined by attachment theory. This specialized neural framework ensures our survival by bonding us to others from the moment we are born. If you find these insights helpful for your bedtime listening, perhaps consider subscribing to follow our deep dive into the mind. John Bowlby, the pioneer of this field, proposed that the attachment system is an evolutionary safeguard, a dedicated circuit designed to maintain proximity between an infant and a caregiver. This is not merely a psychological preference, but a physiological necessity for the developing nervous system. During the first 24 months of life, the human brain undergoes a massive period of synaptogenesis, particularly within the right hemisphere. This growth is heavily influenced by the serve and return interactions between an infant and their primary attachment figure. The orbitofrontal cortex, located just behind the eyes, acts as the primary command center for emotional regulation and social valuation. It matures through the experience of mutual gaze and skin-to-skin contact, which triggers a cascade of neurochemicals like oxytocin and endogenous opioids. These chemicals act as the biological glue that reinforces the bond, creating a sense of internal reward for social proximity. In a secure attachment, the caregiver's ability to consistently attune to the infant's distress facilitates the development of a resilient orbitofrontal cortex. This creates a neural signature characterized by high connectivity between the prefrontal cortex and the amygdala, allowing the individual to modulate their own stress responses effectively in adulthood. When you think back to your own earliest memories of comfort, can you recall a specific sensation of safety that stayed with you? This subjective feeling is the result of the brain's ability to inhibit its own fear centers through established pathways of trust. Conversely, insecure attachment styles leave distinct, observable imprints on brain architecture. Anxious attachment individuals often exhibit a hyperreactive amygdala and a diminished ability for the prefrontal cortex to dampen social alarm signals. This results in a baseline of hypervigilance, where the brain remains in a state of high cortisol production even in the absence of an immediate threat. In contrast, Those with avoidant attachment patterns may show a chronic downregulation of the attachment system. Their brains often suppress the neural pathways associated with seeking proximity, leading to a blunted oxytocin response during social bonding as a protective mechanism against rejection. The biological concept of the safe haven describes the brain's reliance on a trusted figure to downregulate the sympathetic nervous system during periods of high stress. When an adult in a secure relationship experiences a threat, the mere presence of their partner can lower their heart rate and stabilize their blood pressure. This co-regulation is a literal sharing of physiological resources, where one nervous system helps to steady another. Research as recent as 2025 has highlighted how this proximity-seeking behavior is governed by the ventral vagal complex, a part of the autonomic nervous system that promotes social engagement over fight or flight responses. The secure base serves a different but equally vital biological function by providing the neurochemical stability required for exploration and learning. When the brain feels fundamentally secure, it shifts metabolic resources away from survival-oriented limbic processing and toward the dopaminergic systems associated with curiosity and reward. This allows for the high-level cognitive functioning necessary for complex problem-solving and creativity. Longitudinal studies have confirmed that these early blueprints remain remarkably stable into adulthood, though they are not immutable. The brain's inherent plasticity means that earned security is possible through consistent, healthy adult relationships. These connections can physically reshape the orbitofrontal cortex, strengthening the inhibitory fibers that calm the emotional centers. This structural evolution reflects the brain's lifelong ability to learn new patterns of safety and connection. When researchers at Columbia University analyzed the brains of individuals who had recently experienced an unwanted breakup, the fMRI data revealed a striking physiological reality. Upon viewing images of their former partners, the scans showed significant activity in the secondary somatosensory cortex and the dorsal posterior insula. These specific regions are traditionally associated with the perception of physical pain, such as the sting of a burn or the pressure of an injury. To the brain, the loss of a primary attachment figure is not a metaphorical hurt. It is a literal threat to the organism's perceived safety. This overlap suggests that the evolutionary development of the social attachment system likely hijacked the existing physical pain system to ensure humans remained motivated to stay within their protective social groups. It is one of many relaxing facts to consider that our bodies are designed to value connection so deeply that its absence triggers a survival-level alarm. This sensation of physical distress is worsened by a profound neurochemical crash that mirrors the process of clinical withdrawal. During a long-term partnership, the brain becomes accustomed to a steady baseline of oxytocin and dopamine, which act as natural modulators of mood and stress. The sudden absence of these chemicals causes the ventral tegmental area, the brain's primary reward center, to go into a state of hyperarousal. This creates a physiological craving that drives the heartbroken individual to seek out their former partner, much like a person struggling with an addiction. During this period, the levels of serotonin often drop significantly, which can lead to the obsessive-compulsive thought patterns frequently observed in the early stages of a split. Many listeners find that understanding this chemical shift makes bedtime listening a more grounding experience, as it provides a biological explanation for restless thoughts. Beyond the neural pathways, the systemic impact of heartbreak can manifest as a literal, physical change in the heart's anatomy. The endocrine system responds to the emotional trauma by releasing massive quantities of cortisol and adrenaline into the cardiovascular system. In some severe cases, this hormonal flood leads to Takotsubo cardiomyopathy, or broken heart syndrome, where the left ventricle of the heart temporarily changes its shape. This prevents the heart from pumping blood effectively and can cause symptoms that are indistinguishable from a heart attack. It is a powerful illustration of the bidirectional communication between the emotional centers of the brain and the physical health of the body. Do you remember a time when an emotional event caused a physical sensation so strong it felt impossible to ignore? Eventually, the brain begins the difficult task of cognitive restructuring, which involves the dismantling of internal models of the world. Every routine and future plan that involved the partner must be physically unlearned through a process of long-term depression, a neurological term for the weakening of synaptic connections. The hippocampus must work to recontextualize memories, moving them from the active category of daily life into the historical category of the past. While this metabolic process is exhausting, it is a testament to the brain's inherent resilience and the core of science for sleep studies. Slowly, the prefrontal cortex regains its ability to regulate the emotional impulses of the amygdala, allowing the individual to form new neural pathways associated with independence and different forms of social connection. This recalibration is not merely a psychological shift but a physical reorganization of the brain's connective tissue that helps you fall asleep with a sense of eventual restoration. When an attachment figure is suddenly absent, the brain enters a state of profound neurological confusion because it is, at its core, a biological prediction engine. For years, your nervous system has built a complex internal map where the presence of a loved one is a constant, predictable variable. When they are gone, the brain continues to run its established simulations, leading to that jarring, surreal sensation of expecting them to walk through a door or call your name. This is a failure of object permanence at a sophisticated level, where the intellect understands the loss, but the deeper, subconscious circuitry has not yet updated the internal blueprint. The distress following this mismatch is localized in the anterior cingulate cortex, an area of the brain responsible for processing both physical pain and social exclusion. Studies in the field of social cognitive neuroscience demonstrate that this region lights up during grief in the exact same patterns it does during physical injury. This suggests that the ache of loss is not a metaphor but a literal physiological event. The anterior cingulate cortex acts as a neural alarm system, alerting the body that a vital social connection, one necessary for survival in our evolutionary past, has been severed. As this alarm persists, the brain may transition into a state of lethargy often described by the energy conservation theory of grief. This theory posits that the heavy, immobilized feeling of deep sadness is an adaptive response designed to prevent the individual from wasting metabolic resources. If a primary attachment figure cannot be found, the brain forces the body into a low-energy state to ensure survival during the period of transition. It is a biological protective mechanism that prevents the exhaustion of a fruitless search. During these periods of intense quiet, your brain is actually performing incredibly demanding structural work behind the scenes. Have you ever found that your mind wanders back to specific memories of them just as you are about to drift off? This is part of the remapping process, where the hippocampus and the posterior cingulate cortex begin the slow work of updating your internal world map. These structures must physically reorganize to account for the fact that the person is no longer a physical destination or a source of immediate feedback. This involves the pruning of old synaptic expectations and the strengthening of new pathways that recognize the absence. The brain eventually builds a new framework where the loved one is transitioned from a present variable to a remembered one. This doesn't mean the bond disappears. Rather, the neural pathways move from the centers of active expectation to the centers of long-term storage. This remapping allows the prefrontal cortex to eventually regain its regulation over the emotional centers, ending the state of constant alarm. This physical restructuring is what enables the human spirit to eventually stabilize, ensuring that while the map has changed, the ability to navigate the world remains intact. Through this biological evolution, the brain finds a way to carry the memory without the constant physiological stress of the search. When a person enters the threshold of parenthood, the brain undergoes a structural remodeling so profound it mirrors the drastic transitions of puberty. This process is driven by a surge of hormones that trigger a significant pruning of gray matter, particularly within the prefrontal cortex and the temporal lobes. While losing gray matter might sound detrimental, researchers from the Autonomous University of Barcelona have demonstrated that this is actually a process of specialization. By thinning out underused neural connections, the maternal brain becomes a highly efficient machine, fine-tuned to interpret the needs and social cues of an infant. This sculpting is not temporary. MRI studies indicate these structural changes persist for at least 2 years after birth, marking a permanent shift in the woman's cognitive landscape. The amygdala, that ancient almond-shaped center responsible for processing emotions and threats, becomes remarkably enlarged and hyper-responsive during this period. This heightened sensitivity serves a vital evolutionary purpose, keeping a mother in a state of constant vigilance to ensure the safety of her child. It is this neural shift that explains why a mother might wake at the slightest whimper of her baby while sleeping through a loud thunderstorm. The brain essentially redraws its maps of importance, placing the infant at the center of every sensory filter. Have you ever noticed how your own focus shifts so entirely when someone you love deeply is in need of your attention? There is a biological phenomenon known as microchimerism that blurs the lines between two separate lives in a literal, physical sense. During pregnancy, fetal cells cross the placenta and enter the mother's bloodstream, eventually migrating into her tissues and even her brain. These cells are not just transient visitors. They can remain integrated within the mother's neural architecture for decades, long after the child has grown into an adult. This means that a mother literally carries the genetic material of her children within her own mind, a form of biological legacy that persists through time. Scientific studies, including those published in journals like PLOS One, have identified male DNA in the brains of women who had sons years prior, suggesting a cellular connection that is as permanent as it is mysterious. While we often group all deep affection under the single banner of love, the brain recognizes distinct signatures for maternal and romantic bonds. Both forms of love activate the brain's reward system, the rich clusters of dopamine-producing neurons, but they diverge in significant ways. Maternal love specifically activates the periaqueductal gray, a region associated with nurturing behavior and the suppression of pain, which is less prominent in the maps of romantic attraction. Furthermore, maternal love uniquely suppresses the activity of the lateral prefrontal cortex, the area responsible for social judgment and negative emotions. This neural switching off allows for an unconditional acceptance that is rarely mirrored in other human relationships. This specialized circuitry ensures that the grueling demands of early childhood are met with a sense of reward rather than mere exhaustion. The brain's oxytocin system, often called the bonding hormone, works in tandem with these structural changes to create a feedback loop of affection and care. When a mother interacts with her child, the surge of oxytocin reinforces the pruning and the amygdala's vigilance, solidifying the new identity of the maternal mind. It is a transformation that proves love is not merely a feeling, but a fundamental re-engineering of our biological reality. This intricate design suggests that our capacity for deep, self-sacrificing care is written into the very folds of our cortex, ensuring that life continues through a powerful, quiet devotion. While the biological focus often rests on the mother, the paternal brain undergoes a transformation just as profound and chemically guided. When a man transitions into fatherhood, his hormonal profile begins to shift in direct response to the presence of his child. One of the most significant changes is a marked decline in baseline testosterone levels, which often drops by approximately 30% in the first few months after birth. This reduction is not an accidental byproduct of exhaustion, but a targeted biological adaptation designed to dampen aggression and risk-seeking behaviors. By lowering testosterone, the body shifts the father's focus from external competition to internal nurturance, making him more sensitive to the infant's subtle cues. This hormonal recalibration is deeply influenced by the level of physical interaction the father has with his baby. Men who engage in frequent, hands-on care, such as bathing, rocking, or skin-to-skin contact, experience a significant surge in oxytocin, the hormone responsible for bonding and trust. Alongside oxytocin, prolactin levels also rise in responsive fathers. While traditionally associated with lactation in women, prolactin in men is linked to increased responsiveness to an infant's cries and a heightened drive to provide protection. Does the memory of a father's steady presence ever make you wonder how much of that stability was driven by his own changing biology? Beyond the endocrine system, the structural landscape of the father's brain actually rewires itself to accommodate these new responsibilities. Neuroimaging studies published in journals like Nature Communications have shown that the dad brain experiences increased gray matter volume in regions associated with empathy, such as the insula and the prefrontal cortex. These adaptations enhance cognitive flexibility, allowing fathers to better anticipate the needs of a nonverbal child while managing the complex multitasking required for family survival. Interestingly, these biological shifts are not just vital for the child's development but serve as a cornerstone for the pair bond itself. Research indicates a strong correlation between lower testosterone levels and greater marital stability in long-term relationships. This suggests that the same biological mechanisms that facilitate gentle caregiving also work to maintain the cohesion of the parental unit. This interplay between chemistry and behavior creates a feedback loop. The more a father provides care, the more his brain reinforces the neural pathways for patience and devotion. It is a quiet, internal re-engineering that ensures the survival of the next generation through a steady, chemically supported love. This evolution of the male psyche demonstrates that the capacity for nurturing is not a secondary trait, but a primary biological imperative etched into the architecture of the human mind. Beyond the immediate family unit, The architecture of the human mind extends its reach to encompass the complex, non-biological alliances we recognize as friendship. Research published in the Proceedings of the National Academy of Sciences suggests that close friends often share more genetic markers than would be expected by chance. This phenomenon is known as social homophily. It appears that our biology possesses a subtle radar for individuals whose genomes mirror our own, particularly in the regions governing the sense of smell and the regulation of the immune system. Choosing companions with similar genetic profiles may have provided a distinct evolutionary advantage by creating a functional kinship that allowed for more predictable social environments. This genetic mirroring is a silent foundation for the intuitive click we feel when meeting someone who seems remarkably familiar despite being a total stranger. Have you ever noticed how your closest friends seem to share your exact sense of timing or specific sensory preferences? The evolutionary value of these non-kin relationships is rooted in the concept of reciprocal altruism. By cooperating with individuals outside of our direct lineage, our ancestors were able to create safety nets that extended beyond the limits of the nuclear family. This strategic cooperation mitigated the risks of resource scarcity and physical threats, as a wider social circle provided more diverse skill sets and more robust protection. Friendship essentially transformed the high-stakes gamble of individual survival into a collaborative enterprise where the success of the group bolstered the fitness of the individual. The capacity to manage these intricate social networks is not infinite, as it is governed by the physical volume of the neocortex. Anthropologist Robin Dunbar identified a cognitive threshold, often referred to as Dunbar's number, which suggests humans are biologically equipped to maintain roughly 150 stable relationships. This limit reflects the brain's processing power required to track the history, emotions, and shifting loyalties of a specific group of people. While modern digital tools allow us to interact with thousands, our neurological hardware remains tuned to the scale of a small, interconnected tribe. To maintain the integrity of these social bonds, the brain utilizes a sophisticated chemical reward system involving endogenous opioids, specifically endorphins. In our primate ancestors, social grooming through physical touch was the primary method for triggering these feel-good chemicals. Humans evolved to achieve this same neurochemical release through more efficient means, such as synchronized laughter and storytelling. When a group of friends laughs together, their brains experience a simultaneous surge of endorphins that acts as a powerful social glue. This chemical release creates a mild analgesic effect, raising the physical pain threshold of those involved and fostering a deep sense of mutual trust. This internal pharmacy rewards us for being social, ensuring that we remain invested in the companions who provide us with emotional and physical security. Through these biological mechanisms, friendship becomes a vital component of our physiological health, providing a chemical buffer against the stresses of a complex world. Please consider subscribing to continue this exploration of the biological forces that define our humanity. Learning how our bodies reward us for connection reminds us that we are never truly meant to walk through life in isolation. The biological bridge between individuals is built largely upon a specific class of brain cells known as mirror neurons. These specialized cells were first identified in the early 1990s by a research team led by Giacomo Rizzolatti at the University of Parma. While studying the premotor cortex of macaque monkeys, The scientists observed a peculiar phenomenon where certain neurons fired both when the monkey reached for an object and when it simply watched a researcher perform the same movement. This discovery suggested that the brain does not distinguish between self-generated action and observed action as distinctly as once believed. In the human brain, these neural systems are situated primarily within the premotor cortex and the inferior parietal lobe, serving as a functional link between perception and action. When you witness someone expressing a specific emotion, your brain effectively creates a high-fidelity mental simulation of that state within your own motor and sensory circuits. This internal mirroring allows for an intuitive understanding of the intentions and feelings of others without the need for complex deductive reasoning. It is the fundamental biological mechanism that enables us to feel a sharp pang of discomfort when we see someone else trip or experience a sudden, involuntary surge of joy when a partner smiles. This process of shared neural activity is the root of emotional contagion, a phenomenon where moods ripple through a social network with surprising speed and efficiency. Have you ever noticed how a single person's genuine, belly-deep laughter can quickly transform the energy of an entire room? This occurs because our brains are hardwired to subconsciously mimic the micro-expressions, vocal tones, and postural shifts of those around us. By subtly mirroring the physical state of another person, we begin to evoke the corresponding chemical environment in our own nervous systems. Scientists distinguish between two primary forms of this connection: affective empathy and cognitive empathy. Affective empathy is the raw, visceral experience of feeling another person's pain or excitement, driven by the limbic system and the insula. It is an automatic, bottom-up response that provides a direct emotional echo of another's experience. Cognitive empathy, however, is a more analytical, top-down process involving the medial prefrontal cortex, allowing us to intellectually grasp another person's perspective or mental state. While affective empathy lets us feel with someone, cognitive empathy allows us to think about their experience, providing a balanced approach to social navigation and conflict resolution. The strength of this mirroring often depends on the depth of the relationship and the level of attention we pay to one another. Research indicates that we are significantly more susceptible to the emotional contagion of those we trust or admire, as our brains prioritize the signals coming from our inner social circle. Even physiological markers like heart rate and pupil size can synchronize during deep conversation, creating a state of biological resonance that can last long after the interaction ends. This profound interconnectedness suggests that our individual emotional well-being is never truly a solo endeavor, but is instead deeply influenced by the collective state of the people we choose to keep close. The intricate architecture of affection extends far beyond the human experience, manifesting in species whose lineages diverged from ours hundreds of millions of years ago. In the vast expanse of the North Pacific, the Laysan albatross commits to a partnership that can span over half a century. These birds spend years navigating the open ocean in solitude, yet they unfailingly return to the same patch of earth to reunite with a single partner. Their reunions are marked by a complex series of synchronized movements, bows, clicks, and wing tucks that serve to recalibrate their shared bond. Do you remember the first time you observed an animal showing what looked like genuine affection for another? Similarly, the seahorse engages in a daily ritual of connection. Every morning, a pair will meet to change color in unison and swim in synchronized spirals, a behavior that reinforces their commitment and ensures their reproductive cycles remain perfectly aligned. Evolutionary biologists have long puzzled over why long-term pair bonding is so rare among our closest relatives. While we might perceive lifelong devotion as a universal ideal, only about 3 to 5% of all mammalian species practice any form of monogamy. This rarity stems from the intense selection pressures of the natural world, where the drive to distribute genetic material often outweighs the benefits of staying with a single mate. If you could observe any species in the wild for a year, which one do you think would teach you the most about patience? It is essential to distinguish between social monogamy and sexual monogamy when studying these patterns. Many species, including various songbirds and some primates, exhibit social monogamy by co-parenting and defending a shared territory, while simultaneously engaging in extra-pair copulations. This strategy allows for the stability of a shared home while maintaining the genetic diversity required for long-term survival in changing environments. The emotional depth of the animal kingdom becomes perhaps most visible in the presence of death. Elephants are known to exhibit what can only be described as grief rituals, frequently pausing to investigate the remains of deceased relatives. They use their trunks to gently stroke the ivory and bone, often standing in prolonged, heavy silence that suggests a cognitive recognition of loss. How does the idea of an albatross traveling thousands of miles just to find one specific partner make you feel about the scale of the world? This behavior isn't limited to large-brained mammals. The corvid family, including crows and ravens, displays remarkable reactions to the passing of their own. When a crow discovers a fallen member of its flock, it emits a specific call that summons others to gather around the body. While some researchers suggest this is a method of assessing environmental danger, the sheer intensity and duration of these gatherings hint at a social complexity that mirrors our own experiences of mourning. Which of these behaviors feels the most familiar to your own understanding of companionship? In the quiet laboratories where neuroethology is studied, the prairie vole serves as a primary model for understanding the chemistry of devotion. Unlike their cousins, the montane voles, prairie voles form permanent pair bonds driven by a high density of oxytocin and vasopressin receptors in the reward centers of the brain. When these animals mate, the rush of neurochemicals creates a permanent association between the partner and the sensation of pleasure, effectively wiring them for fidelity. This biological mechanism illustrates that what we call love is often a sophisticated survival strategy encoded directly into the nervous system. The biological architecture of affection is not exclusive to our own kind, as evidenced by the profound chemical dialogue shared between humans and dogs. When a person and their canine companion lock eyes, they engage in a unique oxytocin feedback loop that mimics the bonding process between a biological parent and a newborn. A seminal study published in the journal Science demonstrated that this mutual gaze causes oxytocin levels to spike in both the human and the dog simultaneously. It is a remarkable example of convergent evolution, where two different species have developed a shared neurochemical language to ensure mutual protection and companionship. The physiological impact of this bond provides a stabilizing force for the human cardiovascular system. Interaction with a pet is known to significantly reduce blood pressure and heart rate, often more effectively than the presence of a supportive human friend. This occurs because animals provide a form of non-judgmental social support that lowers the production of cortisol, our primary stress hormone. Have you ever found that the silent presence of an animal is more grounding than a complex conversation? Within minutes of petting a dog, the human brain begins to transition from a state of sympathetic nervous system arousal into a parasympathetic state of rest and digest. This connection is facilitated by physical adaptations that have occurred over thousands of years of cohabitation. Domestic dogs have evolved a specialized facial muscle known as the levator anguli oculi medialis, which is almost entirely absent in their wild wolf ancestors. This muscle allows dogs to raise their inner eyebrows, creating the puppy dog eyes expression that triggers an immediate nurturing response in the human brain. By mimicking the facial geometry of a human infant, dogs have effectively hacked the human caregiving system to ensure their own survival and integration into our social structures. This behavior falls under the scientific concept of alloparenting, where the instinct to nurture is directed toward individuals outside of one's direct genetic lineage. In the context of the human-animal bond, this drive crosses the species barrier entirely, creating a biological partnership based on mutual emotional utility. Humans provide resources and safety, while animals provide sensory regulation and social tethering. This ancient alliance has reshaped the human limbic system, making the presence of other species a fundamental requirement for our emotional well-being. The result is a sophisticated tapestry of interspecies love that is written into our very DNA, proving that the need for connection is a universal biological mandate that transcends the boundaries of the human experience. Moving from the broad biological mandate of connection to the specific metrics of human survival reveals that committed partnerships act as a literal shield for the physical body. Long-term epidemiological studies have consistently demonstrated a correlation between marriage and increased longevity, suggesting that the presence of a supportive partner can add years to a person's life expectancy. Researchers have observed that individuals in stable, high-quality relationships tend to survive longer after major medical events, such as heart attacks or invasive surgeries, compared to those who live alone. This statistical advantage is not merely anecdotal, it is a measurable phenomenon documented across diverse populations and age groups over several decades of clinical observation. The mechanism behind this survival advantage often manifests within the microscopic environment of our own immune systems, where social chemistry dictates cellular behavior. High levels of social integration and relationship satisfaction are linked to a more robust immune response, particularly regarding the production and efficiency of white blood cells that target pathogens. People in healthy relationships frequently show enhanced responses to vaccinations, indicating that their bodies are primed to defend against external threats more effectively than those who lack close bonds. When you consider your own social circle, do you find that your sense of belonging changes how physically resilient you feel during the colder months? This biological reinforcement suggests that the feeling of being loved translates into a tangible, cellular form of protection that maintains the integrity of the host. Beyond the immune system, a phenomenon known as the buffer effect serves as a neurological shock absorber against the inevitable pressures of modern existence. When we face acute stress, the presence of a trusted partner can significantly dampen the activity of the hypothalamic pituitary adrenal axis, which regulates the release of cortisol. This co-regulation means that a simple touch or a supportive word can physically lower blood pressure and slow a racing heart rate during a crisis. By sharing the emotional load, the body avoids the wear and tear of chronic weathering, a term scientists use to describe the cumulative damage caused by persistent stress on the cardiovascular and nervous systems. In stark contrast, the absence of these connections can act as a silent biological toxin that alters our very chemistry and accelerates the aging process. Chronic loneliness is associated with heightened levels of pro-inflammatory markers, such as C-reactive protein and interleukin-6, which contribute to the development of chronic diseases like diabetes and atherosclerosis. This systemic inflammation is so pervasive that some meta-analyses equate the health risks of social isolation to the physiological damage caused by smoking 15 cigarettes a day. It appears that the human body interprets a lack of connection as a state of constant environmental threat, keeping the system in a state of high alert that eventually leads to cellular exhaustion and decay. The data suggests that our relationships are far more than social constructs or cultural preferences. They are essential components of our biological architecture. By examining the way our cells respond to companionship, we see that love is a primary driver of physical health and a fundamental requirement for the maintenance of the human machine. This deep-seated need ensures that we remain tethered to one another, not just for the sake of reproduction, but for the day-to-day regulation of our internal systems. These findings emphasize that the presence of a partner provides a unique form of environmental stability that the body relies upon to function at its most efficient level. The neural architecture of this efficiency is governed largely by the amygdala, an almond-shaped cluster deep within the temporal lobes. Within milliseconds of meeting someone, the amygdala evaluates facial cues to determine a baseline of trustworthiness, long before the conscious mind has formed an opinion. This region acts as a sentinel, scanning for subtle inconsistencies that might suggest a threat to one's well-being. When we feel an intuitive sense of safety, it is often because the amygdala has ceased its alarm signaling. Oxytocin plays a critical role in this transition by inhibiting the avoidance instinct. It modulates the neural circuits responsible for fear, effectively lowering the drawbridge of our emotional defenses. By reducing the activity of the amygdala, oxytocin allows the brain to shift from a state of self-protection to one of social engagement. This neurochemical shift is what permits the vulnerability required for genuine connection to take root. True intimacy eventually leads to a phenomenon known as biobehavioral synchrony. This occurs when two individuals become so attuned that their physiological markers, such as heart rate and cortisol levels, begin to fluctuate in tandem. This state represents a profound level of biological trust, where the nervous system of one person becomes an extension of the other's regulatory environment. Have you ever noticed your own breathing slowly match the rhythm of someone you deeply care about while sitting together in silence? Biobehavioral synchrony extends beyond just the heart and lungs. It also involves the synchronization of neural oscillations during communication. When two people trust each other, their brain waves in the gamma and theta frequencies often align, facilitating a more seamless exchange of information. This high-level coordination allows for an intuitive understanding that transcends verbal language. The stability of these internal models is fragile and relies on a consistent history of reliable interactions. When trust is broken, the brain's prediction models undergo a violent recalibration known as betrayal trauma. This isn't just an emotional response. It is a fundamental breakdown in the way the prefrontal cortex processes social data. The brain treats a breach of trust as a critical error in its survival software, often leading to a state of hypervigilance. Neurobiological research indicates that betrayal can cause the anterior cingulate cortex to remain in a state of high alert, as if the body is anticipating physical pain. Because the brain functions as a Bayesian inference engine, it uses past experiences to calculate the probability of future outcomes. A single act of betrayal introduces a massive prediction error that forces the entire system to reevaluate every past interaction. This reassessment consumes enormous metabolic resources, explaining why emotional recovery often feels like a physical burden. Over time, the energy required to maintain this defensive posture can lead to significant physiological exhaustion. Restoring this system requires a slow, deliberate reconstruction of the neural pathways that govern social safety and predictable behavior. Robert Sternberg proposed that the architecture of this social safety is best visualized as a triangle, where the geometry of a human relationship is defined by three distinct vertices: intimacy, passion, and commitment. This framework, refined through decades of psychological observation, remains a cornerstone of behavioral science because it maps so cleanly onto our underlying biological systems. The first vertex, intimacy, represents the emotional core of a connection. It is characterized by feelings of closeness, connectedness, and bondedness that develop gradually through shared experience. In the brain, this manifests through elevated levels of oxytocin and vasopressin, chemicals that facilitate deep trust and effectively dampen the reactive nature of the amygdala. This emotional warmth creates a physiological feedback loop where vulnerability is rewarded with a profound sense of security, reinforcing the neural pathways associated with long-term pair bonding and social stability. The second vertex is passion, the motivational component that drives physical attraction and romantic longing. Unlike the gradual accumulation of intimacy, passion is often rapid and intense, fueled primarily by the dopaminergic reward system. It mirrors the neurochemistry of intense focus, where the presence of the beloved triggers a surge of dopamine in the ventral tegmental area and the nucleus accumbens. This is frequently accompanied by increased levels of norepinephrine, which explains the visceral symptoms of early romance, such as a racing heart, sweaty palms, or a heightened sense of alertness. Have you ever noticed how your perception of physical exhaustion seems to vanish when you are in the early, high-energy stages of a new relationship? This physiological state is metabolically expensive and serves as the evolutionary engine designed to bridge the gap between two strangers, overcoming the natural caution of the social brain. Commitment, the third vertex, functions as the cognitive and decision-based component of the triangle. It involves the immediate decision to love another and the long-term resolve to maintain that connection despite the inevitable challenges of life. This aspect of love resides primarily in the prefrontal cortex, the brain's seat of executive function and rational planning. It is less about the fluctuating tides of emotion and more about the deliberate choice to integrate another person into one's future trajectory. This cognitive stability provides a necessary scaffold, allowing the relationship to remain intact during periods when the other two vertices might temporarily wane. When all three elements, intimacy, passion, and commitment, are present in a healthy equilibrium, Sternberg characterizes the state as consummate love. This is considered the ideal, yet it is a dynamic state that requires constant calibration of these three distinct biological systems. The proportions of this triangle are rarely static throughout the life course of a partnership. In the initial phases, the shape is often heavily skewed toward passion, creating a sharp, narrow profile that lacks a broad base of shared history or long-term planning. As the relationship matures, the passion vertex naturally tends to plateau or decrease in intensity as the brain habituates to the stimulus. Conversely, the intimacy and commitment vertices typically expand, transforming the triangle into a wider, more grounded structure. This evolution is a natural physiological transition from the high arousal state of infatuation to the sustainable, homeostatic state of companionate love. By understanding these shifting ratios, we can view the cooling of initial intensity not as a loss, but as the strengthening of the bond's underlying foundation. This stabilization of the bond is anchored in what psychologist John Gottman describes as love maps, which are intricate mental representations of a partner's inner landscape. From a neurological perspective, constructing these maps involves the hippocampus and the medial prefrontal cortex working in tandem to index and retrieve specific data points about a loved one's dreams, fears, and daily rhythms. Unlike the unpredictable dopamine surges of early infatuation, these cognitive structures provide a reliable framework for navigating the complexities of another person's psyche. As these maps expand, they create a sense of navigational ease within the relationship, allowing partners to anticipate one another's needs with minimal conscious effort. The brain effectively creates a high-definition archive of the other person, ensuring that their preferences and history are always accessible. The brain inherently favors this predictability due to the mere exposure effect, a principle suggesting that repeated encounters with a constant stimulus increase its perceived value and safety. When we see a familiar face, the amygdala's typical vigilance toward the unknown is replaced by a sense of calm, as the ventral striatum releases a steady flow of reinforcing neurochemicals. This preference for the familiar is a fundamental survival mechanism, signaling to the nervous system that the environment is secure and the social bond is stable. By reducing the cognitive load required to assess a partner's intentions, the brain can redirect its resources toward other complex tasks. This is why the presence of a long-term partner can physically lower blood pressure and cortisol levels during times of high stress. Memory consolidation plays a pivotal role in this process by weaving individual moments into a cohesive, shared narrative identity. As time passes, the brain's encoding of personal history becomes inextricably linked with the presence of the partner, a phenomenon that researchers have linked to increased relationship satisfaction through the overlap of neural representations. This shared story is not merely a collection of facts, but a neural integration where the boundaries between the self-concept and the partner concept begin to blur. Can you remember a specific moment when your partner's reaction became so predictable that it felt like an extension of your own thoughts? This overlap is often referred to as self-expansion, where the resources and perspectives of the partner are integrated into one's own mental model of the self. Beyond the emotional benefits, knowing a partner's habits results in significant energy conservation for the brain. The theory of predictive coding suggests that the brain is constantly trying to minimize prediction errors about the world. In a long-term relationship, the high level of familiarity allows the brain to form accurate internal models of the partner, which drastically reduces the metabolic cost of social interaction. We no longer need to exert high levels of executive function to interpret subtle facial expressions because the baseline of the partner has been firmly established. This efficiency allows the relationship to function as a low-energy, high-reward system, providing a sustainable foundation for long-term health and psychological resilience. This cognitive ease transforms the complexity of two lives into a single, harmonized rhythm. This psychological evolution is often described as the Michelangelo phenomenon, where partners act as sculptors of one another's personalities and behaviors. Just as the artist claimed to see the ideal figure trapped within the stone, supportive partners reinforce the traits that align with a person's own ideal self-image. Over years of interaction, this persistent affirmation leads to measurable shifts in identity, as the brain's neuroplasticity allows it to reshape its self-concept based on the feedback of the beloved. This process suggests that we are not fixed entities, but rather dynamic works of art influenced by the hands of those we choose to keep close. As these lives intertwine, the couple begins to develop a transactive memory system, essentially merging two individual minds into a single, high-capacity database. This cognitive division of labor means that one partner may instinctively store information about social obligations or family history, while the other retains technical knowledge or spatial directions. By offloading specific types of data to one another, the individual cognitive load is lightened, allowing the pair to solve complex problems with a collective efficiency that neither could achieve alone. Do you find yourself relying on someone else to remember the small details of your shared history? The brain physically restructures itself to accommodate this long-term partnership through significant changes in gray matter density. Longitudinal neuroimaging studies have observed that long-term couples often exhibit neural synchronization, where their brainwave patterns align during conversation or shared activities. This synchronization is particularly evident in the prefrontal cortex and the insula, areas associated with empathy and high-level decision-making. These physical adaptations are a testament to the brain's ability to treat a partner not as an external object, but as a fundamental component of its own operational landscape. These structural changes provide a powerful defense against the natural vulnerabilities of the aging process. Strong social bonds and the daily cognitive demands of a long-term relationship contribute to a robust cognitive reserve, which helps delay the onset of dementia and other neurodegenerative conditions. The constant stimulation of navigating a shared life acts as a form of neurological exercise, keeping the brain's executive functions sharp and resilient. Research from the Harvard Study of Adult Development indicates that the quality of our social connections is one of the strongest predictors of cognitive health in our later decades. The neurochemical environment created by a stable, loving connection further supports this biological longevity. Frequent positive interactions maintain high levels of oxytocin and vasopressin, which promotes synaptic growth in the hippocampus, the brain's center for learning and memory. This hormonal balance also helps suppress the production of cortisol, the primary stress hormone that can damage neural tissues over time. By reducing chronic inflammation and protecting the integrity of the blood-brain barrier, a deep emotional bond serves as a biological shield, ensuring that the mind remains as vibrant as the connection that sustains it. This biological shield is not a luxury reserved for any specific civilization or era, as romantic love has proven to be a near-universal human experience across the vast landscape of global history. Extensive ethnographic research, including a landmark study of 166 diverse cultures, identified the presence of romantic love in 89% of those societies. In the few instances where it was not explicitly documented, researchers often found that the omission was a result of limited sampling rather than a genuine absence of the emotion. This suggests that the neurocircuitry for deep attachment is a fundamental hardware component of the human brain, rather than a software program installed by modern media or Western literature. While the internal chemical surge remains remarkably consistent, the outward vocabulary of affection varies wildly depending on local customs and traditions. In many Western cultures, the romantic narrative focuses heavily on the individual's pursuit of intense, passionate euphoria as the primary catalyst for long-term commitment. In contrast, many collectivist societies traditionally view love as a secondary outcome that grows from a foundation of shared duty, family alignment, and social stability. This does not mean the feeling is absent. Rather, the cultural script dictates that love is the result of a successful partnership rather than the prerequisite for one. For some, this raises an interesting question. Do you feel that a shared sense of purpose is more grounding than the initial spark of physical attraction? Scientists have observed that while the catalyst for entering a relationship changes across borders, the long-term physiological benefits of a secure bond, such as lowered blood pressure and enhanced immune function, remain constant regardless of how the couple first met. The specific gestures we use to signal our devotion are also products of our environment. While the romantic kiss is common in many parts of the world, it is not a universal human behavior. Several cultures in sub-Saharan Africa and the Amazon historically viewed the practice with confusion or even distaste. Some societies express deep intimacy through the gentle rubbing of noses or the sharing of breath, demonstrating that the biological drive for closeness will always find a path through the available cultural medium. Even the concept of the one is a relatively modern, localized construct that would be entirely foreign to many historical nomadic tribes where communal bonds were the primary focus of emotional security. Jealousy provides perhaps the most striking example of how biology and culture interact to shape our feelings. Evolutionary psychology suggests that jealousy evolved as a survival mechanism to protect reproductive investments and ensure the stability of the family unit. However, the triggers for this intense emotion are highly modulated by the values of a specific society. In cultures that place a high premium on paternity certainty, physical infidelity often triggers the most severe physiological stress response. Conversely, in societies where social and economic resources are shared more broadly, emotional betrayal or the diversion of time and attention often causes greater distress. The brain's amygdala reacts to the perceived threat, but the cultural environment determines exactly what looks like a threat in the first place. Through this lens, we see that love is a universal biological melody that every culture arranges with its own unique instruments and rhythms. The intentional acceleration of intimacy was famously catalogued in 1997 by psychologist Arthur Aaron, who sought to determine if closeness could be engineered within a laboratory setting. This landmark study at Stony Brook University moved beyond the observation of existing couples to see if strangers could develop a deep bond in under an hour. The primary mechanism used was a series of 36 questions, meticulously structured into 3 distinct sets. These questions follow the psychological principle of reciprocal self-disclosure, a process where individuals gradually trade increasingly personal information. By starting with lighter inquiries, such as describing a perfect day, and ending with profound reflections on mortality or personal failures, The participants bypass the superficial layers of social interaction. This method works because reciprocal self-disclosure creates a feedback loop of trust and validation. When one person takes a social risk by revealing a vulnerability, and the other person meets that risk with their own honesty, the brain interprets this as a signal of high-level safety. While many people fear that showing weakness will lead to repulsion, the science of social bonding suggests the opposite is true. Vulnerability acts as a form of social currency that demonstrates a person's willingness to be perceived as they truly are, which in turn reduces the perceived threat to the other individual. Within this framework of bedtime listening, we can see how the reduction of social anxiety through structured honesty allows the nervous system to shift from a state of hypervigilance to one of profound calm. The experiment concluded with a final, nonverbal task: 4 minutes of silent, mutual gazing. This specific duration is significant because it exceeds the typical length of comfortable eye contact in casual conversation, which usually lasts only a few seconds. Prolonged eye contact triggers a physiological phenomenon known as neural coupling, where the brain activity of the two individuals begins to mirror one another. This state of synchrony allows the participants to feel a sense of shared identity and deep recognition that often bypasses the limitations of language. Incorporating these relaxing facts into your understanding of human behavior can provide a sense of clarity about why we feel drawn to certain people. As you utilize this science for sleep, consider how the architecture of intimacy is built not on grand gestures, but on the steady, rhythmic exchange of truth. The 36 questions remind us that the biological drive for connection is hardwired into our neurochemistry, waiting for the right conditions to be activated. By understanding the mechanics of self-disclosure and the power of the gaze, we gain insight into the very foundation of how humans create the social structures that sustain us through the quietest hours of our lives. This selfless extension of the self into the well-being of others finds its most profound biological expression in the concept of agape, or universal compassion. While romantic love often centers on the neurobiology of reward and pair bonding, altruistic love engages a sophisticated network of brain regions dedicated to social cognition and empathy. When an individual acts with pure concern for another's welfare, the brain suppresses the self-referential processing of the medial prefrontal cortex. This shift allows for a state of consciousness where the boundaries between the self and the other become porous, a phenomenon long observed by neurologists studying the meditative states of long-term practitioners of compassion meditation. Deep within the brainstem, the periaqueductal gray matter serves as a critical node in this caretaking architecture. Traditionally known for its role in modulating pain, this region is also a primary driver of the maternal instinct and nurturing behavior across mammalian species. When we witness the vulnerability or distress of another creature, the periaqueductal gray activates, signaling a reflexive urge to provide protection and care. This primitive circuit ensures that the impulse to nurture is not merely a high-level intellectual choice but a fundamental survival mechanism hardwired into our oldest neural structures. Research published throughout 2024 and 2025 has clarified how this region interacts with the oxytocin-rich hypothalamus to transform a perception of need into a physical drive to assist. The neurological reward for such behavior is often referred to as the helper's high, a tangible state of euphoria following an act of charity or kindness. This experience is the result of the mesolimbic pathway's activation, where the ventral striatum and nucleus accumbens release dopamine in response to prosocial actions. Interestingly, the brain often registers the act of giving as more rewarding than the act of receiving, suggesting that our neurochemistry is biased toward generosity. This internal reinforcement system helps sustain the social cohesion necessary for complex civilizations to flourish, turning the abstract virtue of kindness into a repeatable, pleasurable biological event. From an evolutionary standpoint, the existence of such selflessness presented a significant puzzle for early theorists. If natural selection favors the survival of the individual, why would an organism sacrifice its own resources or safety for another? The answer lies partly in kin selection, formalized by Hamilton's rule, which posits that altruism can evolve if the cost to the giver is lower than the benefit to a relative who shares their genetic material. By protecting our kin, we are effectively protecting the persistence of our own DNA into the next generation. Do you remember a moment when you helped a complete stranger and felt a sudden, quiet warmth in your chest? This sensation is the byproduct of reciprocal altruism, a strategy where humans cooperate with non-relatives based on the implicit understanding that help given today may be returned in the future. This complex social exchange requires advanced cognitive abilities, including the capacity to remember faces, track reputations, and detect cheaters who take without giving back. Through these mechanisms, the evolution of altruism has transformed the competitive landscape of nature into a collaborative network where the success of the group becomes the success of the individual. Modern intimacy has transitioned into a digital theater where the ventral striatum serves as the primary neurological engine for our social interactions. This specific region of the brain, integral to the dopaminergic reward system, reacts to every notification and screen-based interaction with a measurable pulse of gratification. Data suggests that these frequent, low-stakes digital rewards create a feedback loop that fundamentally alters our baseline expectations for emotional intimacy. Neuroimaging studies finalized in 2025 demonstrate that the intermittent reinforcement provided by social media notifications mirrors the neural patterns observed in classic behavioral reinforcement cycles. Such patterns can lead to a state of hyperarousal where the brain remains in a constant state of social scanning, perpetually waiting for the next digital validation. While these screens provide a sense of near-constant availability, they often fail to facilitate the essential biological process of physical co-regulation. In a traditional face-to-face setting, our autonomic nervous systems engage in a subtle, silent dialogue, synchronizing heart rates and balancing cortisol levels through nonverbal cues. This physiological mirroring is a cornerstone of human bonding, yet it is noticeably absent in text-based or even standard video-mediated communication. Research published in Nature Neuroscience within the last year confirms that the absence of tactile feedback and pheromonal exchange in digital environments limits the release of oxytocin. This neuropeptide is vital for the development of long-term pair bonding and deep-seated trust between individuals. Have you ever noticed how the quality of your calm differs after a digital conversation compared to a quiet moment shared in physical proximity? The evolution of AI companionship has reached a profound milestone as we navigate the early months of 2026. We are observing that the human brain is increasingly capable of forming parasocial bonds with generative entities designed to simulate high levels of emotional intelligence. These systems are trained on extensive data sets of human empathetic responses, allowing them to offer consistent validation that triggers genuine emotional reactions in the user. However, findings from longitudinal studies in late 2025 indicate that while these digital interactions can mitigate the immediate symptoms of loneliness, they lack the limbic resonance necessary for building long-term emotional resilience. The prefrontal cortex may recognize the interaction as artificial, yet the emotional centers of the brain often respond to the simulated empathy as if it were a tangible human connection. This duality creates a complex psychological state where the user feels heard but remains physiologically isolated. Digital dating platforms have introduced the paradox of choice to the landscape of modern romance, creating a significant cognitive load for the individual. When the brain is presented with a theoretically infinite pool of potential partners, the executive functions can become overwhelmed, often leading to a state of decision paralysis or reduced relationship satisfaction. A 2025 study from the University of Oxford's Department of Experimental Psychology found that high-volume browsing environments can actually desensitize the ventral tegmental area. This desensitization results in a diminished capacity to feel a strong emotional spark, as the brain begins to categorize potential partners as replaceable data points rather than unique biological beings. This shift suggests that our ancient evolutionary hardware, designed for the intimacy of small group survival, is currently struggling to adapt to the boundless, rapid-fire choices offered by the digital age. We are learning that while technology can bridge the physical distance between us, the biological requirements for deep connection, scent, touch, and rhythmic synchronization remain deeply tethered to our physical presence. The mathematics of commitment reveals a calculated threshold for when to stop searching and start choosing. This principle is often referred to as optimal stopping theory, a concept derived from the classic secretary problem in probability studies. To maximize the likelihood of finding the best possible partner, the math suggests observing and rejecting the first 37% of potential candidates in your dating window. After this point, you should commit to the very next person who surpasses the quality of everyone you previously encountered. This strategy effectively balances the need to gather enough data to understand your options with the risk of waiting so long that the best candidates are no longer available. Perhaps you remember a moment when you realized your standards had shifted because of someone you had met previously? Beyond the initial selection, researchers use differential equations to model how a couple's interactions evolve over time. Dr. John Gottman and mathematician James Murray spent years refining a model that predicts the longevity of a marriage with over 90% accuracy. Their findings emphasize the 5-to-1 ratio, which dictates that for every negative interaction during a conflict, there must be at least 5 positive ones to sustain a stable bond. When this ratio shifts toward a 1-to-1 balance, the relationship enters a mathematical state of decay known as a downward spiral where negative feelings become self-reinforcing. It turns out that the most successful couples are those with low negativity thresholds, meaning they address small annoyances immediately rather than letting them accumulate into complex, unsolvable variables. The mechanics of trust can be further analyzed through game theory, specifically the iterated prisoner's dilemma. In a single encounter, it might seem logical to protect oneself by acting selfishly, but in the repeated rounds of a long-term relationship, the most successful strategy is tit-for-tat with forgiveness. This involves starting with cooperation, mirroring your partner's positive actions, and occasionally forgiving a lapse to prevent a permanent cycle of retaliation. By choosing to subscribe to this cooperative framework, partners minimize the mathematical cost of conflict and maximize their collective payoff. When we consider the statistical probability of finding a soulmate, the numbers can feel daunting. In 2010, mathematician Peter Backus applied the Drake equation, originally designed to estimate the number of alien civilizations in the Milky Way, to his own love life. He found that out of the millions of people in London, only 26 were statistically likely to be a suitable match for him based on his specific criteria. While these odds seem slim, the math of human connection is often saved by the law of large numbers and the fact that humans are not static variables. We possess the unique ability to influence the probability of our own success by expanding our social networks and refining our communication, turning a game of pure chance into one of calculated, beautiful intent. This ability to adapt ensures that while the numbers provide a framework, the human element provides the solution. This internal solution leads directly to the most profound challenge in modern neuroscience, often referred to as the hard problem of consciousness. While we have spent decades mapping the intricate dance of neurotransmitters, we still face a significant gap between biological function and subjective experience. We can observe the precise moment oxytocin floods the brain or track the specific neural pathways activated by the sight of a loved one, yet these data points do not explain why it feels like something to be alive and in love. Philosophers and cognitive scientists use the term qualia to describe these individual instances of subjective, conscious experience. Think of it like describing the color blue to someone who has never seen it. You can explain the physics of light waves and the biology of the human eye, but the actual sensation of blueness remains private and uncommunicable. In the context of our emotions, qualia is the raw, felt texture of a heartbeat skipping or the specific, heavy warmth of a shared glance. It is the what it is like aspect of our existence that escapes even the most rigorous chemical equations. As of early 2026, despite our ability to monitor brain activity with incredible resolution, the bridge between objective matter and subjective mind remains uncrossed. We have the map, but we are still learning how to read the legend. Scientific inquiry into the nature of the self suggests that while we can identify the biological correlates of affection, the internal narrative of the heart is not yet something we can fully digitize or replicate. This limitation is not a failure of science, but rather a testament to the staggering complexity of the human condition. Recognizing that love is a product of evolutionary biology and chemical signaling does not make the feeling any less real or significant. Understanding the mechanics of a sunset, from atmospheric refraction to the physics of light, does not diminish the beauty of the horizon. In the same way, acknowledging that our deepest attachments are rooted in survival instincts and hormonal surges provides a framework for understanding, but it does not replace the emotional reality of the bond. We find ourselves in a unique position where we can appreciate both the clockwork of the brain and the poetry of the mind, accepting that the mystery of how one creates the other is part of what makes our experience so profoundly human. Everything we have explored, from the nanosecond firing of a synapse to the decades-long stability of a partnership, illustrates a seamless continuum of biological engineering. We began with the frantic, adrenaline-fueled rush of attraction, a state where the brain temporarily mirrors the obsessive patterns of clinical disorders, yet we conclude with the quiet, restorative presence of companionate love. This transition is governed by the shifting ratios of vasopressin and oxytocin, molecules that literally reshape the architecture of our neural pathways to accommodate the presence of another. It is a remarkable journey from the microscopic to the macroscopic, proving that the most complex social structures in our world are built upon the humble foundations of carbon, hydrogen, and nitrogen arranged in precise, elegant sequences. While the biological imperative is to protect the genetic line, the human experience translates these chemical signals into a sense of profound purpose. To the amygdala, love may be a safety mechanism or a proximity alert. However, to the prefrontal cortex, it is a narrative of shared history and future goals. This duality does not diminish the value of our feelings but rather enhances them, showing that nature has found a way to make survival feel like transcendence. Research in epigenetics even suggests that these deep bonds can influence gene expression, dampening the body's inflammatory responses and promoting cellular repair. We are not merely puppets of our hormones, but active participants in a feedback loop where our cultural choices and personal values influence the very biology that drives us. In a broader sense, these feelings represent a moment where the universe, through the medium of biological life, begins to value itself. When you feel a deep connection to another person, it is a manifestation of the same fundamental forces that hold atoms together, scaled up to the level of sentient consciousness. We are the stars looking back at their own light, finding warmth not just in the fusion of hydrogen, but in the proximity of another mind. It is the ultimate expression of complexity that a collection of atoms can feel a sense of belonging to another collection of atoms, creating a loop of meaning that defies simple entropy. As you settle into the quiet of the night, consider how these intricate systems continue to work within you, maintaining the silent, invisible bonds that define your existence. If you find these scientific explorations a helpful companion for your bedtime listening, I invite you to subscribe so we can continue this journey through the wonders of the natural world together. Understanding the mechanics of our hearts and minds only makes the experience of being alive more extraordinary. May your curiosity remain as resilient as the bonds we have studied, and may your rest be as deep as the mysteries we have yet to uncover.